right, everybody, I just wanted to quickly record this lecture so that you guys have the opportunity to hear me lecture and try to make this as fair as possible since we are doing the lab virtually. So today I'm going to be talking to you about cardiovascular physiology. So let's go ahead and get started. So as you guys know, these are the assignments somewhat of what you're going to be doing. So just be aware that one is an online activity that you will have to and turn into Sakai. This is one that you can do at home by yourself. Blood pressure, we will certainly have the opportunity to do that one in the next couple of weeks. But there's a good video that I want you guys to watch. And then for the ECG, I would like for you to read the section about the ECG and also, excuse me, complete procedures 1 and 2 in your lab meeting. Okay. So we're going to first talk about auscultation, and auscultation just means the listening of heartbeats using a stethoscope. And so there are 4 areas primarily that you will auscultate when it comes to listening to different, specifically different valves, because as we know already, the heart sounds are made. We already know that heart sound come from closing of valves. So our S1 heart sound, or lub part, comes from closing of our AV valve, and our S2, the dead part, comes from closing of our semilunar valves. So there are 4 areas that you listen to whenever you are using a stethoscope to listen to the heart sounds. And those 4 areas, they coincide with the 4 valves. So we have the aortic area. This is where you'll best hear the sounds coming from the aortic valve. The pulmonic area, the tricuspid area, and also the mitral area. When it comes to listening to these different sounds, you're doing so through a stethoscope, and understand that a stethoscope actually has 2 different parts. So it has a diaphragm and a bell. And so what I mean by this is that if you are looking at a stethoscope, you'll notice there's a bigger circle, and then there's usually a smaller circle. Okay, so these 2 different circles correspond and can be used to listen to either high-pitched or lower-pitched sounds. So the bell used to hear lower-pitched sounds is the smaller piece in the middle that's kind of raised up off of the larger diaphragm, which is used to hear higher-pitched sounds. Right, so going on to understanding some different concepts when it comes to auscultation. First and foremost is heart rate. You can determine heart rate by listening to these sounds, and heart rate is the number of beats per minute that you hear. So normally this should be between 60 and 100 beats per minute, also known as tachycardic. To be considered tachycardic, usually this is a rate that is greater than 100 beats per minute. And also bradycardia, that refers to a heart rate that is fewer than 60 beats per minute. Now, when it comes to rhythm, this is going to be the pattern and the regularity of the beat. So what it should be, it should sound like lub-dub, lub-dub. However, if we're talking about an irregular rhythm, it might be lub-dub, 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 lub-dub. There's not a good normal interval between each lub-dub, and that's what irregular rhythm would mean. If there is what we call an irregular rhythm, that is the term arrhythmia. Arrhythmia simply means irregular rhythm. And additionally, some sounds that you might hear that are not completely normal are things like a murmur, and this would sound like a click or a swoosh between heartbeats or between heart sounds. And some of the reasons for this would be things like regurgitation. And if you guys remember, regurgitation is just backwards flow of blood. So instead of going from the, let's just say, for example, instead of going from the right ventricle out the pulmonary trunk, now we go from the right ventricle back into the right atrium, and that would be blood that's being regurgitated. It's moving backwards. Okay. Stenosis is another reason that you might hear some abnormal sounds or some swooshing sort of sounds. And stenosis just means that the vessel or chamber or the valve has become more narrow. Okay, so basically what I mean by this is that if this is a normal vessel, sometimes what can happen is there's a section that just became more narrow, and maybe that's because perhaps if this is regular, perhaps there's been some plaque buildup, and now there's just less area for the same amount of blood to travel through, so you'll hear like a swoosh sound. Okay. So now I'm going to play a short video of some different heart sounds and heart murmurs that I would like for you guys to sort of follow along with and listen to. It's really interesting that you can actually hear these things. So let's go ahead and play some of this video.
When a healthy heart beats, it makes a lub-dub sound. The first heart sound, lub, also known as S1, is caused by the closing of the AV valves after the atria have pumped blood into the ventricles. The second heart sound, dub, or S2, originates from the closing of the aortic and pulmonary valves right after the ventricles have ejected the blood. The time interval between S1 and S2 is when the ventricles contract, called systole. The interval between S2 and the next S1 is when the ventricles relax and are filled with blood, called diastole. Diastole is longer than systole, hence the lub-dub, lub-dub, lub-dub. Heart sounds are auscultated at 4 different sites on the chest wall, which correspond to the location of blood flow as it passes through the aortic, pulmonic, tricuspid, and mitral valves, respectively. This is how similar defects associated with different valves are differentiated. Heart murmurs are whooshing sounds produced by turbulent flow of blood. Murmurs are diagnosed based on the time they occur in the cardiac cycle, their changes in intensity over time, and the auscultation site where they are best heard. Examples of conditions associated with common systolic murmurs include mitral valve regurgitation, when the mitral valve does not close properly and blood surges back to the left atrium during systole. The murmur starts at S1 when the AV valves close and maintains the same intensity for the entire duration of systole.
So obviously you can very much in that section hear the difference in the sounds between our very 2 distinct S1, S2 sounds versus mitral regurgitation, where it just sounds like the sound has been kind of combined.
This hollow systolic murmur is best heard at the mitral region, the apex, with radiation to the left axilla. On the other side of the heart, a tricuspid valve regurgitation has similar timing and shape, but is loudest in the tricuspid area, and the sound radiates up along the left sternal border. Aortic valve stenosis. When the aortic valve does not open properly and blood is forced through a narrow opening. The blood flow starts small, rises to a maximum in mid-systole at the peak of ventricular contraction, then attenuates toward the end of systole. This results in a crescendo-decrescendo, or a diamond-shaped murmur, which starts a short moment after S1. It is often preceded by an ejection click caused by the opening of the stenotic valve. Aortic stenosis murmur is loudest in the aortic area, and the sound radiates to the carotid arteries in the neck following the direction of blood flow. Again, on the other side of the heart, a pulmonic stenosis has the same characteristics, but is best heard in the pulmonic area and does not radiate to the neck. Other conditions that cause audible systolic murmurs include ventricular septal defect,
So I just wanted to play that video for you guys and let you hear the differences between a normal and an abnormal R sound. All right, so moving on to the next topic is vascular exams. So vascular exams basically tell us how healthy the blood vessels are. And there's a lot of different ways to assess this, one of them being pulse palpitation. Obviously, if there is a missing pulse in an extremity or in some location, that would mean that there's an obstruction of blood flow that we would need to restore. Also, you can auscultate vessels and listen for what we call bruits, which are these swishing, blowing sounds that are caused by turbulent blood flow. And turbulent blood flow, the best way that I think I can describe it is sorry if you hear my dog barking. He sees deer in our backyard and he's not happy about it. So anyways, back to bruits. The best way for me to describe bruits is kind of to compare it to a windy day. And you know, when it's blowing like crazy, kind of goes maybe something like that, and you can hear like a whistling sound. It's sort of the same thing in blood vessels. So imagine that we put blood in a blood vessel, and because there is maybe some, just draw like some plaque buildup maybe. So if there is some plaque buildup, then as the blood is flowing through the vessel, sometimes if there's like a plaque buildup, what happens is the blood vessel, the blood cell is going to run into this plaque. And when it does that, it kind of bounces off of it and has to find a new route. So that's where you get that sort of wind-like sound, so like this blowing, swishing sound. That's really just the best way for me to describe it. Hopefully that makes sense to you guys. I'm going to erase this and get this out of the way. So the other way that we can assess how healthy our blood vessels are is by doing a really simple, what we call capillary refill test. And capillary refill, by definition, is going to be the time that it takes for capillary beds to refill after they have forcibly been emptied by pressure. So for example, we have capillaries in our fingernails, in our nail beds. So if you press down on your nail and you wait until your nail looks pretty much all white, and then you let go, you'll notice that it starts to turn pink again. So what you've done is when you've pushed down and the nail is all white, you have forcibly removed all of the blood from the nail. And then when you let go, the pink is just the blood coming back into those capillary beds. And normally it should take between 1 and 3 seconds for the nail to return to normal. And this is just assessing all of the vessels basically proximal to that particular nail bed. So if we're talking about our fingernails, that would assess the health of the vessels from basically the tips of our fingers all the way up to the aorta, if you will. And so if this takes longer than 1 to 3 seconds, then maybe as healthcare professionals, we can order some sort of test that will assess those blood vessels, maybe some sort of imaging or what have you, that can assess the blood vessels that are proximal to that capillary bed and see if there's a problem somewhere. Okay. So our third topic is blood pressure. And blood pressure, by definition, is going to be the pressure exerted on the walls of the blood vessels. And so what this means is I'm going to just draw a quick little picture here. Is if this is a blood vessel, inside of our blood vessels, we have, of course, our red blood cells. And because it's a pretty packed situation inside of our blood vessels, the blood actually pushes against the walls of the container that they're in, or in this case, the vessel. Just like if we had, do another example here. If we were looking at some sort of flask, if we're looking at some sort of flask and we put some water in there, the water is pushing against flask walls, right? We can see that the water would be pushing against flask walls. Sort of the same situation over here in the blood vessels. The blood is going to push, because it's the fluid, it's going to push up against the walls of the container that is holding it. And this, how strong of a force gets pushed up against those walls is what we're calling blood vessels. Excuse me, blood pressure. Oops, sorry about that. I'm trying to erase here. Pen's not working for me. There we go. And blood pressure depends on 3 different things. So the strength of the blood pushing against the walls is really dependent on 3 things. So it's dependent on cardiac output and also peripheral resistance. And then the third thing that's not really in this equation, but it sort of can be sort of intuitive, is blood volume. So looking back at that drawing that I did, vessel, if there's more blood inside, then there's going to be a higher pressure against these walls, right? Because there's just more substance in this closed container. Whereas if there is less blood, so in this situation our blood is like really full, if there's less blood, then there'll be some pushing against the walls, but perhaps not as much. Okay. So cardiac output is going to be equal to stroke volume times heart rate. Peripheral resistance is basically going to have to do with diameter of the blood vessel. And then that, when we multiply those things together, you get blood pressure. So let's talk about cardiac output and peripheral resistance in a little bit more detail. So cardiac output, by definition, because I like to give you guys definitions here, cardiac output is the amount of blood pumped by each ventricle in 1 minute. And normal value for this is between 4 and 8 liters per minute, which is basically equal to our blood volume. Because in the human body, we have normally about 5 to 6 liters of blood. And so we can see that our heart is circulating our entire blood volume every single minute, which really, if you stop and think about it, is quite incredible. Again, cardiac output mathematically is equal to our heart rate multiplied by our stroke volume. And so heart rate is simply a measure of how many times the heart beats every minute. And normally this is between 60 and 100, which we talked about earlier. And then stroke volume is how much blood the ventricle pumps out every time that it contracts. And this should be between 60 and 100 milliliters. So every time the heart beats, it is forcing out 60 to 100 milliliters of blood. Peripheral resistance, as we know, affects cardiac output because I said it in the last slide. But basically, this is the impedance to blood flow that is encountered by the blood vessels themselves. Remember I said it has to do kind of with diameter, is one possible way to think about it. So for example, vasoconstricting, or basically this means the blood vessels are getting more narrow. Vasoconstriction is going to increase resistance. And resistance, think about it like resisting blood flow through that artery or that vessel. So when the vessel gets smaller, it's resisting that blood flow even more than normal. When it gets bigger, then quite the opposite is happening. There's going to be less resistance. Obstructions like plaques and etc. You can kind of think about obstructions or plaques as being a sort of vasoconstriction, because if there is an obstruction or a plaque, So let's draw one on here. That section, as I kind of referred to this earlier, basically from here to here now, we're seeing that our diameter is smaller than normal. So it's kind of like vasoconstricting the vessel in that way. Now, as I mentioned already, we have a lot of discussion here with peripheral resistance and diameter, and kind of continuing on with that. Resistance is highest away from the heart in the periphery. And hopefully this makes sense because when we look at the heart and the aorta, so as the aorta comes off and it gives rise to all of these different vessels, all of these little teeny tiny vessels and all these little tiny capillaries, the size of the vessels decreases. So the resistance is going to increase because the size of the vessels decreases the farther you get from the heart. And hopefully that makes sense as well. So we can clinically evaluate blood pressure using instruments such as a sphygmomanometer and a stethoscope. So the sphygmomanometer is basically just a fancy word for a blood pressure cuff. And a stethoscope. So whenever we are using a blood pressure cuff, as you're seeing here, we know that this part inflates, and then this little handle is what makes it inflate. And then we put a pressure gauge on there to see the pressure of the cuff. And then you take a stethoscope and you put it either on the brachial artery or the radial artery. In this case, it's on the brachial artery just before it splits into the radial artery and the ulnar artery. So essentially what you're doing is as you inflate this cuff, you are cutting off blood flow, right? You're totally stopping blood flow from this point downwards. Okay, so the first time that you do this, and I'm going to teach the way that you're supposed to do it for nursing and for the NCLEX is you're going to put your stethoscope, well, first of all, you're going to palpate. First, you're going to palpate this blood vessel. So you're going to feel for a pulse right there in the crease of the elbow. Once you have found that pulse, you are going to inflate the blood pressure cuff. So you're going to squeeze on this and inflate the blood pressure cuff and watch the pressure gauge. So keep inflating the cuff until you no longer feel a pulse. When you no longer feel a pulse, you're going to take a peek at your pressure gauge, and whatever it reads, when you do this for real, you want to go an additional, usually like 20 to 30 millimeters above what that pressure gauge said. Okay. So then you let it go, and now you're going to do it for real this time. So the first go round is actually feeling the pulse and feeling the absence of the pulse. That gives you kind of a starting point of how high you need to inflate your cuff in order to get an accurate reading. So you deflate the cuff, and now you place your stethoscope and your pressure gauge, and you start pumping it up to whatever number you decided to go to based on that initial reading. And then you start slowly, using this little dial, releasing the air. As you release the air, we're now deflating the cuff. Blood flow is going to start being restored to the vessels down here, right? Because you've cut off blood flow and now you're releasing it, and now blood flow is going to start coming back. When you hear blood flow coming back, it's going to produce what we call Korsakoff sounds. And because you have squeezed so hard on that vessel, which is kind of what this picture is representing, so you have cut off the blood flow, that's going to create this sort of turbulent sound, this sort of swooshing sound that we call Korsakoff sounds. All right, they're created by these pulse-like blood flow through that compressed artery. So the first sound that you hear is going to represent the systolic pressure. And this is the pressure that is in the arteries when the ventricle contract. And so in this case, this would be the pressure in the artery as the left ventricle is contracting and shooting blood out the aorta. Okay, so it's going to be a pretty high pressure. It should be anyways, which makes kind of sense. Normal, this should be between 100 and 120 millimeters of mercury. Then you're going to keep deflating the cuff, deflating the cuff, deflating the cuff until you don't hear any more sounds. So keep an eye on this pressure gauge, because the last beat that you hear before you don't hear any more sounds is going to be our diastolic pressure. Diastolic pressure. Okay, and the diastolic pressure represents the pressure in the arteries when the ventricle is relaxing or in diastole filling with blood. And normally this should be between 60 and 80 millimeters of mercury. Then you take these two numbers and as you probably have seen before, blood pressure is written as, oops, it's a bad right here, maybe 120 over 80 or 100 over 60. Anywhere in this range is considered normal. If you go below this value, we're talking about hypotension. Anything below this, anything above this, we're talking about hyper. Okay. All right. Erase all this stuff. And we will move on. All right, so the last topic that we'll be talking about is the electrocardiogram. And here we're going to switch from talking about blood pressures and volumes to talking about electricity and electrical signals. So in the heart, we have two different types of cells. We either have pacemaker cells, and this constitutes about 1% of all the different cell types in the heart, or we have contractile cells. And contractile cells is AKA cardiac muscle cells. And about 99% of the cells in the heart are these cardiac muscle cells. But back to this weird pacemaker cell thing. The pacemaker cells are kind of like neurons in that they are electrically excitable. The difference is that pacemaker cells are intrinsic electrically excitable cells, meaning they don't require nerves to stimulate them. They fire on their own. So, with that being said, the cardiac conduction system consists of the SA node, which is going to be in the upper right atrium, and this is our primary pacemaker. This pacemaker, these pacemaker cells depolarize, or they send action potentials 60 times per minute on average. So really, for lab, I'm going to say 60 times per minute, but it can be up to 75 times. But for lab and for the exam for lab, know that SA node is about 60 times a minute. So the SA node is going to generate action potentials that spread to the right atrium, all throughout the right atrium. Also, you can see over to the left atrium. And via this internodal pathway, the AV node is going to receive the signal from the SA node. Okay, the AV node is what we're going to call our backup pacemaker. And we call it a backup pacemaker because if for some reason the SA node stops working, say maybe you have a heart attack and this is the area that gets affected by the heart attack, so this is the area of dead tissue, then the AV node can take over as the pacemaker. The only difference being that the AV node pacemaker cells only depolarize, or they send action potentials at 40 times per minute. So if the AV node has to take over, this is what you're going to see in the patient. They are going to have a slower heart rate compared to normal. Okay, so we have the SA node firing its action potentials and then reaching the AV node. Important note, very important note here. There is a delay when the action potential reaches the AV node. Why? The reason for this is because the SA node depolarizes or sends signals to the contractile cells in the atria and stimulates them to contract. The AV node delays the signal in order to allow the atria to finish their contraction before the signal then gets sent down what we call the bundle of His and the left and right bundle branches, which will conduct the signal down through the interventricular septum, as you can see, and then the signals are going to sort of turn around and travel up the ventricles. And as they're traveling up the ventricles, they are depolarizing the muscle cells in the ventricles, thus stimulating the ventricles. Oops, excuse me, sorry about that. Stimulating the ventricles to contract and thus sending blood out. Their respective areas, okay? All right, so let me erase this stuff. We will move on to talking about what this has to do with the electrocardiogram, or ECG, or EKG. Okay. All right, so the electrocardiogram records that pathway that we were just talking about, the SA node to the AV node, to the bundle of His, to the left and right bundle branches, to the Purkinje fibers. The electrocardiogram records this information. if there is no electrical activity going through the heart, that is why we see a flat line on an ECG. Okay, that means there's no electrical activity. The heart is no longer pumping, it is not working. The flat line that occurs between waves is basically the pacemaker cell action potential is occurring. So it's essentially the flat line, for example, here, and the flat line, for example, here. This is when the muscle is contracting. So I'll talk about what that means. So let's first look at these different waves. So we have the P waves, which is this first wave, the P wave. This is when the SA node depolarizes the left and the right atria. Okay, so depolarization of the left and the right atria. Following depolarization comes contraction. So after the atria have been activated, they're going to contract. This segment that you see here, it's flat because there's no longer electrical activity passing through here. Now this is when the atria are actually contracting, okay? Which is going to force blood down into the ventricles. All right, the QRS waves, this is the set of 3 waves that's representing the depolarization of the ventricles. So at this point, this is when the AV node has received the electrical signal from the SA node and sends that signal down the left and the right bundle branches and back upwards through the Purkinje fiber system. This is ventricular depolarization, okay, or activation of the ventricles. The QRS duration, how long this should take, should be about 0.12, less than, equal to 0.12 seconds. Okay. Now, as we can see, just like after the P wave or after the activation of the atria, we also have another flat line between there and there, right? This is going to represent when the ventricles are actually contracting and sending blood out the aorta or the pulmonary trunk. Okay, so that is what this represents, this flat line. And now, after the ventricles have contracted and they are relaxing again, or they're repolarizing, we see this T wave. And so this is ventricular repolarization. So the final wave that represents, or the recovery wave, when the ventricles are now starting to relax again. So a normal heartbeat, you can see the EKG strip over here. So ECG or EKG, they mean the same thing. Okay, so this is a normal heartbeat. You can see all of the different waves, the P wave. So here we see the P wave. Here's our QRS wave. Here is our T wave, followed by a P wave, QRS wave, T wave, P wave, and so on, right? In a fast heartbeat, you'll notice that each beat is much closer together. And I know that because when I'm looking at these small little squares here, so let's say that there's approximately, what, 3.5 or so, 3.5 squares between this wave and this wave. Here, there's only 2 between those 2 waves, which means that this heart here is beating faster. Here, there's many different boxes. I don't even know, 1, 2, 3, 4, 5, 6 boxes between each beat. That would mean the heart is beating pretty slow. And then earlier when I was talking about lub dub, lub dub, lub dub, lub dub dub dub, lub, right? That irregular heartbeat, meaning that each beat is not at normal intervals or at equal intervals. So what I mean by that is, like, if we look at these two beats, there's 3 boxes in between them. If we look at the one right before that, there's 1, 2, 3, 4.5 boxes between those 2 beats. The beat right before that has, if you add those together, maybe 2. So these are not at regular intervals, okay? Clear off my drawings, and we will on to the next one. So I was kind of just showing you a little bit about the idea of these intervals. And intervals are just the segments or the number of boxes between waves. So first is this R to R interval, and this is the duration and of the generation and spread of the action potential through the heart. Okay, so R to R interval is how long between each heartbeat. How long between each heartbeat. Okay, and this is actually used to determine heart rate. So the normal value, normally it should be between 0.6 and 1 second between each heartbeat. So what I mean by this is, get my out. I then do another. The time that it's between this one and the one right after is our R to R interval. And the next couple of slides has a little bit of a better idea of what I'm talking about. But here's R to R interval for now, okay? The P to R interval is going to be the period from the beginning of our wave. So beginning of our P wave, which we can see here, to the beginning of the QRS complex. And basically what this one's going to represent then is the depolarization from the SA node spread throughout the atria until the AV node receives the signal. So SA node until AV node and all of the actions that occur between. This includes the AV node delay, which lasts about normally AV node usually delays the signal about 1/10 of a second. And so that means that to send the signal from the SA node throughout all of the atria until it receives the AV node, until the AV node receives it, is really taking, since the AV node is delaying by 0.1 second, is really taking 2/100ths to 1/10 of a second to do all of that. It's just incredibly fast if you think about it. Next is the QT interval. And the QT interval is the period from the beginning of the Q wave to the end of the T wave. So this is representing the ventricles both depolarizing and repolarizing. And that's obviously going to include them contracting and relaxing. So normally this takes between 0.42 and 0.44 seconds. So in this manner, we can kind of see that as we're sort of adding these different values together, if we're looking at PR plus QT, this whole thing is taking between, what would that be? 0.54 to 64. Okay, this whole spread throughout the entire heart is taking between 1/2 and 6/10 of a second. That is super, super fast when you really just sit and think about that for a second. Okay. All right, so I just mentioned the RR interval, and I'm going to finish the lecture talking about why that is relevant and how that's useful. So what we have here is an EKG strip. And so when someone's getting an ECG or an EKG, it gets printed on this graph paper, and this graph paper is really useful because it can help you determine some different values. And so kind of going back to that, I'm just going back one slide. So each large box is equal to 2/10 of a second. Each small box is 0.04 seconds. So 0.04, 0.04, 0.04, 0.04, 0.04. So that equals a large box, which is 2/10 of a second, and then, sorry, 5 large boxes, 1, 2, 3, 4, 5 large boxes is 1 whole second, right? Because 2/10 times 5 is a whole second. So every 5 boxes is a second. Now, if every 5 boxes is 1 second, then every 30 boxes should be 6 seconds, right? So if we have 1, 2, 3, 4, 5, 1, 2, 3, 4, 5, 1, 2, 3, 4, 5, 1, 2, 3, 4, 5, 1, 2, 3, 4, 5. Okay, if that is 1 second, that's a second, that's 1 whole second, that's 1 second, that's 1 second, that's 1 second. All of this together is equal to 6 seconds. So I have 30 large boxes here. They're equal to 6 seconds. This is really useful because what you can do is you look at how many total wave sets you have in this segment of 6 seconds, and you can quickly determine heart rate by doing this. So for the best way kind of to do this is to look at the R waves, which are these really tall ones, because those are usually super easy to see, but just make sure that you have a complete wave set. So we have 1, 2, 3, 4, 5, 6, 7. Okay, so we have 7 beats. So if we have 7 beats in 6 seconds, that's equal to how many beats in 60 seconds? Well, this is really easy because 6 times 10 equals 60. So 7 times 10 is 70. And since 60 seconds equals a minute, this is going to be equal to 70 beats per minute. 6-second rule, super easy way to determine heart rate from an ECG, right? Okay, I'm going to give you 2 other methods. So the 6-second rule, although it's really fast, it's not always perfectly precise, which can make it a challenge. But this one's really good for determining or heart rates. You have an irregular ECG or an irregular rhythm. this is a really good way to determine rate when the rhythm is irregular. Here we have a regular rhythm, but if this rhythm were irregular, the 6-second rule is the most common way to determine heart rate from an irregular rhythm. Here are the other two ways to determine heart rate from an ECG. So we'll start with the 300 method. And so here what we're going to do is we're going to look at one R to R interval. And for all intents and purposes, I'm just going to look at this one here. Okay, so I'm going to look at this one. And this one goes from here to here. Okay. Now what I'm going to do is I'm going to count the number of big squares between those R to R intervals. So I chose these ones because they're pretty darn close. So we have 1 big square, 2 big squares, 3 big squares, 4 big squares, and it's like a fifth of a big square. So we'll say 4.2, right? Because 0.2, 0.4, 0.6, 0.8, and 1. So we're saying 4.2 big squares. So if I do 300 divided by 4.2, gotta get out my calculator, and you guys can get out your calculator too. 300 divided by 4.2 is actually equal to 71.43, or approximately 71 beats per minute. Notice how this way is a little bit more precise because this is the same EKG strip as on the last slide. And on the last slide, we said it's 70. Here, we're saying it's 71. You can also do this same thing, but this time you can use small squares. So instead of doing all of these big squares, we can count the number of small squares that we have. And so we have, I know there's 5, there's 10, there's 15, there's 20, about 21 small squares, right? Because there's 1, 2, 3, 4, 5. 1, 2, 3, 4, 5, that's 10. 1, 2, 3, 4, 5, that's 15. 2, 3, 4, 5, that's 20. And 21. Right, so here we're going to do 1500 divided by 21. And we're going to get 71.43, aka 71 beats per minute. Okay, so this is how you can determine heart rate based on EKGs or ECGs. And if you have any questions, please feel free to send me an email. But that is all that I have for you guys today. So go ahead and go through those different activities that I have set up for you for lab in the online world, and watch different videos, and let me know if you guys have any questions. Have a good day.